Hi everyone, welcome to Chapter 8, which is all about cell-cell communication. So the devotion for this week is from Philippians 4, 6 through 7. Do not be anxious about anything, but in everything by prayer and petition, with thanksgiving present your requests to God. And the peace of God, which transcends all understanding, will guard your hearts and minds in Christ Jesus. So this gives us permission and in fact a request to do devotionals to really bring things in prayer to God. Communication with God is key. Communication is key in many things, including our own lives here on earth, communication with friends, with family, and obviously communication with God through prayer, bringing our concerns, our joys, our thanksgivings to God. All right, so let's talk about chemical signaling though. So here we have two coyotes. Given the conditions of their body, one of these two coyotes should likely have a greater appetite than the other. Right? So hopefully it's fairly obvious that this one should feel hungrier than this one. But this is not a trivial thing. We take it for granted, but the idea of hunger has to come from our brain, but it's based on what's happening in all our fat cells in our stomach and things throughout the body. So how does all this aspects get communicated? Right? Well, it's through chemical signaling. So it's known that the fat cells produce a chemical that sends a message to the brain. So the fat cells produce leptin. And if you have a lot of fat cells, the cells are producing a lot of leptin, and then that travels through the bloodstream. It's a hormone, and then that communicates with the brain and causes the brain to act differently. And so in this case, the leptin sends signals to the brain that causes decreased overall feeding because this coyote is very healthy, plenty fed. In this coyote, which is starving, there are very few fat cells left. Therefore, the chemical signal leptin is very low, and so there is none of that signal being sent to the brain of this coyote, and this coyote is consistently hungry, which makes sense because it has the desire to feed because it is starving. So, if that leptin is a chemical messenger, in this case it's a protein hormone. So if it's a protein, we know that there must be a gene that codes for the sequence of that protein, because the sequence of leptin gives it the fold, which we know gives it the function. So there must be a gene for that chemical message, and if so, that gene might be capable of mutating. So if that gene is changed, then the sequence of our protein leptin will be changed, and this will cause leptin maybe to not work. And in fact, there are a breed of mice that basically has this. These are called OB mice for obese, or DB for diabetic, but there are mice that are just constantly hungry. And so this is the wild type mouse, this is the OB mouse, and so we know that this is genetic, and therefore a gene, because it's passed on, it's a trait passed on to the offspring. And so one of the questions is, why is it obese? And one of the hypotheses that can be that the obese mutation results in the inability to produce a functional, correctly folded, appetite-suppressing hormone leptin. And the way people have tested this in the past is through a process called parabiosis. So basically, they fuse the bloodstream of these two mice together. And they've done it more recently just basically by consistently taking blood from a wild-type mouse and kind of transfusing it into the OB mouse. But let's think about what's happening here. If this mouse is producing a normal functioning leptin, then that normal functioning leptin will end up in the bloodstream of this mouse and should also suppress the appetite of this mouse. And indeed, that's what happens. So after several weeks of this parabiosis, the OB mouse, despite still having that genetic mutation, ends up being a normal size because its appetite is suppressed appropriately by that gene leptin. So this is cell communication. The fat cells are communicating through a leptin protein hormone that travels through the brain stream, communicating with the brain, and then the brain is basically communicating with the rest of the body through other signals that induce or decrease appetite based on the amount of leptin signal it received. So the conclusion here would be that the instructions for making the chemical signal leptin are carried in a gene. We know that if that gene is mutated, that can lead to a non-functional leptin that, at least in the case of the OB mice, is what is causing the lack of appetite suppression. And we also know that this chemical signal, this hormone, travels through the blood and therefore affects the appetite. So in this case, leptin is the signaling molecule, and the appetite response is the responder. So what are the main steps in cell signaling? Well, basically the first thing that has to happen is the ligand, the chemical signal, has to bind to a receptor. So this is a receiver of that signal. The ligand binds to the receptor, and that causes an interaction. And this usually happens at the cell surface. We'll talk more about this throughout this chapter. That then leads to a signal transduction. So there is often a cascade of molecules on the inside of the cell that takes that signal from the cell surface, where the receptor is, to the part of the cell that is going to ultimately respond, and that leads to the response. So here's kind of the idea of that. I apologize for the graphics here. I'm sure they're beautiful. But we have a signaling molecule. When that signaling molecule binds with a receptor, that has to cause a change in the cytoplasmic side. So think about this for a second. The receptor crosses the membrane. This signaling molecule usually is polar. Not always, but usually is polar. And since it's polar, it can't get through the membrane on its own. We learned all about this in chapter 5. Right? So it has to bind to the receptor on the membrane, and something about this receptor has to change on the inside of the cell to help the inside of the cell realize that the ligand is bound on the outside. So the change, the binding on the outside of the cell, has to cause a conformation change to the intracellular portion of this transmembrane domain that allows it to be viewed as a change. Then you have the signaling cascade, multiple signaling molecules transferring the signal, and finally the cell will change appropriately in response to whatever that signal is giving. So don't pay attention too much to the details here, but this is kind of an example of a time when a cell would need to respond on the inside to something on the outside. All right, so here we have a bacteria in a salting environment. And some bacteria have cell walls, some bacteria have two membranes. So in this case, salt is starting to pour in and being at a very high concentration between these two membranes. And we know that if you are in a non-isotonic solution, osmosis can happen. So this hypertonic solution can actually cause water to go out of that cell, and this bacteria cell would shrink if it doesn't do something about it. So somehow it has to sense that the surrounding environment here is too salty, and it has to make an appropriate change. So there are receptors in the inner membrane here. So this receptor crosses the membrane and can sense this saltiness on the outside. And when it becomes too salty, that causes a change on the intracellular side. In this case, it becomes phosphorylated. That then activates another protein, a signaling cascade molecule that previously was not turned on. The phosphate group basically turns it on and now activates it. And this goes and causes transcription of a new gene. So now in response to this extra saltiness, too salty, the gene is turned on, making mRNA, to make this protein that then can go and block this channel that is allowing salt to come in. So now this channel is blocked, thus preventing this from getting even more salty and basically protecting the bacteria from death. So a signal on the outside of the cell led to a change in the receptor, a change in a cascade molecule, a change in gene expression, and the response in this case was to create a protein that wasn't being made before to block salt from entering between the two membranes. Right. So, but this leads to a really important question. We say that the surrounding environment is too salty. Well, how does the receptor differentiate between salty and too salty? And this is really important because some salt is important. If it's not salty enough, you certainly don't want to create something to block salt coming in, because as bad as hypertonic solution is, hypotonic, not enough salt, is just as bad. So how can the receptor sense from some saltiness to too salty? And this idea is threshold. All right, so a driver returned to his car after a 3-hour shopping spree to see the following sight. His car would not start. Why is that? What's your hypothesis for why this car would not start? Well, many people often say the battery is dead, but that's not the case. Why is the battery, how do we know the battery is not dead? Well, the lights are on. It's just that the battery does not have enough juice to turn the car on. So in that case, we sometimes say the battery is dead, but it's not completely dead. It's just doesn't have enough juice to start the car. So what I want you to do is think about how the battery ignition threshold would look on this graph. So basically we have low battery current, high battery current, then we have no ignition and ignition. So go ahead and just on a piece of paper draw this out. What do you think is going to happen? Well, a lot of people kind of draw a straight line. So as the battery current goes up, we go from no ignition to ignition. But that would indicate that you can have partial ignition. You can't really have partial ignition. It's all or none. The car either starts or it doesn't. So the ignition really looks more like this. When you don't have enough battery current, you don't get the car started. And then suddenly there's a threshold, there's a certain amount of battery current that's needed, and the car will start. And maybe there's a little bit of a slope here because there's a point where the car can, or trying, but for the most part it's all or none. And this is an idea of threshold because you either have enough battery current to start the car or you don't. So what do you think the threshold plot for the headlights would look like? All right, so here I've given you three options, A, B, and C. So B we know is the threshold. That's the plot for the battery. So the question is, would the threshold for the lights look like A compared to B? Would it be the same as the battery, or would it be over here like C? Well, we know that the threshold for turning on the lights is lower. You don't need as much battery juice to turn on the lights. So therefore, we don't need as much battery current for the lights to go on. That would be A, because the threshold is lower. We had enough juice for the lights, but not enough juice for the battery. The threshold for lights was lower than the threshold for the battery, or for the car starting. So A here would have the lowest threshold because you need the least amount of battery current to start it. B has a middle threshold, and C would have the highest threshold on these three graphs. And since the threshold for the starting car is B, we know we need less battery current for the lights. So my next question for you is, what happens when you see this? What do you think's happening in your body? Well, hopefully your adrenaline starts rising, right? So your fight or flight response is coming on. And so if you're a more brave person than me, you might fight that bear. If you're more like me, you're going to run and just hope the person next to you is slower runner than you. But either way, adrenaline is going to hit you fast, right? But let's think about adrenaline for a little bit in terms of threshold. So we know that adrenaline has different effects. This same ligand has different effects on many different tissues in the body, right? So adrenaline will cause your pupils to dilate. This allows you to see better for the kind of fight or flight response, right? You get dry mouth often if you're a little bit anxious. Stimulates glucose release. So glucose is released from your liver so that you can have energy for the muscles to respond very quickly in that fight or flight. Right? You constrict blood vessels in the skin, so the very periphery of your body, you constrict blood vessels. And this is actually an evolutionary trait because you don't want to bleed out. If you're in that fight with the bear and the bear cuts your arm, you don't want to bleed out. You want to kind of save that blood for your central vital organs in the body. Airways are relaxed so you can breathe better. Your heart rate speeds up, again part of the fight or flight response, kind of stimulates sweating. But not all of these happen at the same threshold. Just like the battery had different thresholds that was needed to turn on the lights versus the amount of battery that was needed to turn on the car itself, there's different amounts of adrenaline that will turn on these different things. And that's important, right? So you're going to have a lot of adrenaline for when you see that bear up close, right? And you want that to cause a really rapid heart rate, all these things at once for that fight or flight response. But there are other things that cause adrenaline spikes or even just anxiety. All right, so if you're out on a first date, you might have a little bit of sweaty palms, a little bit of nervousness, even dry mouth. But thank goodness that doesn't really cause your blood vessels to constrict in the skin and your heart rate to go way, way fast and things like that. That is a different level of adrenaline that causes different responses. All right? When you're sitting down for an exam, hopefully your anxiety doesn't go to the same extent as fighting a bear. And so only a few of these things will happen with low levels of adrenaline. So for example, your dry mouth, that might have a lower threshold. You get dry mouth with less adrenaline spike than it takes to cause your heart rate to go really rapid, your airwaves and glucose release and so forth. So how does this relate? Well, adrenaline, this chemical signal, also has a threshold response, right? And it's going to have a certain amount of adrenaline that's required to cause your heart rate to spike, but it might have a different threshold for dry mouth or sweaty palms. So the adrenaline can lead to different responses in your body based on threshold. There's a certain amount needed to cause an effect. Just like there was a certain amount of salt needed to hit the threshold to cause that bacteria to start making those proteins that block the salt channels. So my next question for you, which of the following graphs represents a signal with the lowest threshold? A, B, C, or D, and give an explanation of why or how you know. Well, remember, threshold refers to the amount of ligand, the amount of signal needed to cause a response. And so the one with the lowest threshold would need the least amount of signal to cause a response. So A here has the lowest threshold, and D has the highest threshold because you need the most ligand to cause the response. So A might be those things like sweaty palms or dry mouth with adrenaline. D might be blood vessel constriction to the periphery or really rapid heart rate or things like that that needs a really high level of adrenaline to cause it. So there are two things that a cell can control for altering the threshold, and this is important because we really don't want the threshold for dry mouth and sweaty palms to be the same as for heart rate. Even worse, we don't want kind of everything to cause an adrenaline spike as if we were fighting a bear, even if we're just sitting down for an exam or something like that. So what controls this level of threshold? Well, one is the number of receptors on the cell surface, and second is the type of receptor and how strongly it binds. Alright, so I'm going to do my best here to show this in a demonstration. So here I have my example of a ligand, right? And here I have my example of a receptor on the cell. In order for that signal to happen, the ligand has to bind to the receptor and stick. Alright, so while that ligand is bound to the receptor, that signal can happen in the cell and cause a response. So if this is adrenaline, and adrenaline is flowing through your bloodstream and it binds to a receptor in your body, that receptor can cause a response. And maybe this is a receptor in your palms, and that response is sweat. Or maybe it's a receptor in your eye, and the response is dilation. Or maybe it's a receptor in the liver, and the response is glucose release, and so forth. But either way, the ligand has to bind to the receptor. So let's think about this with regards to threshold. If I have a limited amount of ligand, and I want to get a response, would I want more or fewer receptors if I want that threshold to be low? So let's think about this for a second. You have one chance to throw this ball and get it into the cups. Alright, so we're talking about a carnival type game. You have one chance to throw the ball and get it into these cups. Alright, so if I said if you get the ball into the cups and it stays in the cups, you get an automatically A on the next exam. Which are you going to aim for? Are you going to aim for the single cup over here, or are you going to aim for the multiple cups here? Well, which one has a better chance of you getting the response you want, ball in cup, with the least amount of throws? Well, hopefully it's pretty obvious that you're going to aim for the one with many cups, because with the same amount of ligand, one throw, I have a better chance of getting this ligand to stick in the cups if I have a lot of different cups. And so if a cell has a lot of receptors, it is going to be able to have the ligand bind to the receptor, and it's going to have that signal at a lower threshold. So the first one here is the number of receptors on the cell surface. And here is the take-home message. Take this down. The more receptors a cell has, the lower the threshold will be. Because if you have a lot of receptors, you have a better chance of a small amount of ligand finding one of those receptors and causing a signal. Just like you have a better chance of winning that carnival game and getting your automatic A on the exam if you throw that ligand, throw the ball into a whole bunch of cups versus just trying to aim for one. So more receptors, lower threshold. All right. The second part of this is the type of receptor and how strongly it binds. This one's a little bit harder to demonstrate, but here I have some bigger cups. All right, I have bigger cups, and I have the smaller cup. Now, affinity is not, at the cellular level, is not just a measure of the ability of that ball to get in. It's the ability of the ball to stay in. Because remember, you need that ligand to bind to the receptor and stick. So in this case, this would be an example of high affinity. A high affinity ligand and receptor combination binds very, very strongly, right? All those interactions between the ligand, the hydrogen bonds, and the hydrophobic effect, and the ionic bonds, and everything between the ligand and the receptor are very strong here. So once it binds, it's not going to fall off. In this case, this would be an example of a low affinity receptor. So it can bind, but it also is going to fall off pretty easily. Right? So let's think about this for a second here. So the type of receptor and how strongly it binds. If the affinity goes up, in other words, the tighter the binding, the lower the threshold. Because if you're able to bind tightly, now once that ligand is bound, it's not going to let go, and you're going to have a signal with less ligand. So the two things that affect threshold are the number of receptors, higher receptors, lower threshold, and the affinity, tighter binding, lower threshold. So let's think about this with regards to what we've been talking about again. Here's another question for you. So which graph represents a cell with a receptor with the highest affinity? Alright, so remember that high affinity means lower threshold. So D here has the highest threshold. So it would have very low affinity. That would be an example of something that binds very weakly. That ligand, even if it binds it, is probably going to fall off. So you would need a lot of ligand to make sure it keeps binding. Let's talk about A and B now. So these seem to have a lower threshold because you need less ligand. A is a bit of a strange one because its response starts up here. This is not the response to the ligand, this is just the innate response. So no ligand will still have A's response be like this. So what you're looking at is the inflection, where it turns. So this is actually A's threshold, someone here, meaning B has the lower threshold. Right? So B would represent the receptor with the highest affinity because it has the lowest threshold. High affinity, low threshold. Right? So what I want you to do now is just write a two-sentence explanation about the relationship between affinity and threshold. So as affinity goes up, threshold does what and why? Well, hopefully, as you know by now, as the affinity goes up, as that binding is tighter between the ligand and receptor, the threshold will go down because you don't need as much ligand to cause a response. It's going to stick to that receptor pretty tightly. Right? So as the affinity goes up, threshold goes down because the binding is tight. Ligand will be very attracted to that receptor and stay stuck. What about the relationship between the number of receptors on the cell and the threshold, and why? Well, same idea. The more receptors that are on that cell, the better chance that ligand, whatever amount of ligand you have, has to find one of those receptors and bind. So the higher the number of receptors, the lower the threshold. So A would have the highest number of receptors, therefore giving it the lowest threshold. D, the lowest number of receptors, the highest threshold. All right, so how does this relate again back to adrenaline? Well, when you see this, your adrenaline levels are going to spike, right? But some things happen at the lowest level of adrenaline. So things like the production of saliva, dry mouth, if you will, the sweaty palms, etc. Those are going to have a lower threshold. You don't need as much adrenaline spike for that to happen. So the receptors that cause lack of saliva production, the receptors that cause sweating in your palms, those are going to have high number of receptors, therefore a lower threshold, and they're going to have high affinity, also therefore lower threshold. However, things like constricting blood vessels in the skin, you don't really want that to happen until you have a really, really high level of adrenaline. You don't want that to happen just sitting down for an exam. So those responses have a relatively low number of receptors, and the affinity is low. That gives it a high threshold. Now, that only happens if you have a really high level of adrenaline. And that's how this one ligand, this one signal, adrenaline, can have so many different effects on so many different parts of the body, and those effects can happen at different thresholds, at different levels. Right? What happens when you see this? All right, so you've beaten that bear down, you turned and fought it, you beat it, it's walking away in shame, congratulations. What happens? Well, chances are good your adrenaline rush doesn't go away immediately. That adrenaline high is going to stay for a while, and that's because the threshold goes backwards too. As you gain adrenaline, you're going to eventually hit the threshold where you get that fight or flight response. But chances are good your adrenaline spiked way up here. So after you've defeated that bear, it takes a little while for the adrenaline to go away. So you're still on that adrenaline high until adrenaline drops to this level, and now your threshold goes away. So just like when you buy a new battery, you have a lot of juice up here. It takes a number of times for you starting your battery for that battery to lose its juice to the point where your car will no longer start. It'll take a while for your adrenaline to go down to the point where you feel back to normal after fighting that bear. So this idea of threshold is really important in biology, right? So for example, there's a threshold with your immune system. Your immune system wants to attack and kill the cells that are infected, say by COVID-19. That's how you can be protected from things like viruses. If that cell is infected with the virus, the T cells, the killer cells in your immune system, want to kill that cell to protect the rest of your body. But you want that to happen only at a certain threshold. You don't want all your T cells just killing every cell in your body. That's autoimmunity. You also don't want the threshold to be too high where you can get really, really sick before your immune system even starts to take things and basically protect yourself from infection. So you want that threshold to be just right where suddenly it sees a certain amount of infection and it really acts, kills that cell. Right? There's thresholds with regards to hormones in development. There's thresholds with regards to environment sensing. We already talked about the bacteria sensing if it's in too salty an environment and so forth. Well, that happens in our cells as well. Maybe not necessarily with salts, but certain other things. There are thresholds of signals from other cells. At some point, our cells will respond. Right? And so what is that response? Well, there's 3 stages of cell signaling. Right? The stage 1, again, is ligand binding to receptor. And that receptor has to have a change on the intracellular side. Right? So this is the cytoplasmic conformation of the receptor when there's no ligand bound. This is that conformation change when there is a ligand bound. This allows the inside of the cell to be able to see, okay, receptor is like this, there must be no ligand bound. Oh, receptor is like this, ligand must be bound, it's time to send the signal and do a response. Right? So step 1, ligand binds to the receptor, and the receptor changes conformation. Okay. So that's kind of what we were talking about there. Whoops. Ligand without the hormone bound, ligand with the hormone bound, and now it's activated. That will cause a response. Right. So once that response happens, you need a signaling transduction. Right. So this receptor is in the membrane, and that's on purpose because it has to bind to the ligand on the outside of the cell and signal the inside of the cell. But that means it's stuck on the membrane. It can't go and send that signal to the response. So it needs messengers along the way. So signal transduction is when protein messengers kind of send the signal. This guy activates this one then activates this activates this, and then ultimately, step 3, well, that will lead to the cellular response. And maybe it's activating enzymes that weren't active before, maybe it's changing the structure of the cell, or maybe it's turning on new genes, making new proteins that the cell needs in this new environment based on that new signal. So, the last thing I want to end with is the 5 types of cell communication mechanisms. Right? So what do you think the mechanisms are by which cells communicate with each other? See if you can look some up. So look up methods of cellular communication, 5 types of cellular communication. See what you find. Just try and write a few of these down. We'll go through all of these. What types of signals, ligands, can they use? Well, most of the signals are actually protein signals, but there can be some hydrophobic signals like steroid hormones, things like that. But most of them are going to be proteins that are signaling a cell. So the ligands are usually proteins. How can that signal be passed from one cell to another? Well, one cell will make that signal ligand and secrete it, therefore releasing it, and then it will bind to a receptor on an adjacent cell. Therefore, the cell has released the signal, right? That's like me talking. I'm releasing the signal, and that ligand goes to the next cell, like the sound travels to your ear, and you receive the cell, and hopefully you're learning because of that. So one cell can release the ligand, secreted protein, and then another cell has the receptor and binds to it and will respond. All right, so 5 modes of signaling. One is direct intercellular signaling. The key thing to know about this one is that this is through gap junctions. And we briefly learned about gap junctions before, but gap junctions basically are channels that connect two cells together. So the gap junction isn't a channel from the inside of one cell to the outside of the cell. It's from the inside of one cell directly to the inside of another cell, right? And so these can be channels kind of between two cell membranes. And what that means is if I have a cell signal that causes ions or some type of signaling in this one cell, that signal will immediately travel from high to low to the next cell. So both of them will respond at the same time. One of the best examples of this is your heartbeat, right? You have muscle cells, but you want to coordinate the beating of those muscle cells so that your contraction is happening with all the muscle cells at once. That way the blood gets pushed from the atrium into the ventricles, and then the ventricles beats all those muscles all contract at once to send that blood throughout your body. If the muscles were contracting at different times, that's afibrillation, and that's really not pushing blood throughout the body. That's a problem. All right, so how does that happen? How do we get that coordination? Well, there are gap junctions between the muscle cells, and there is a neuronal signal that basically tells the cells when to contract. That's telling your cell when your heart muscle cells when to beat. And so when that signal is there, it's going to cause calcium to flood in, and it's the calcium flooding in that causes the muscle contraction. You don't need to know the details of this, but this is kind of the interesting part. So when calcium floods into the muscle cell, it contracts. But calcium is an ion that can flow very rapidly through channels. And so if one muscle cell gets calcium in it, it's going to flow immediately through gap junctions to all the adjacent ones, and all those muscle cells are going to beat at once. And so your heart beats together in coordinated fashion with all the muscle cells contracting together. And so if you have a second, go ahead and you can even just look up heart muscle cells contracting in culture, and you can see even cells growing in culture, if they're cardiomyocytes, they will beat at the same time. They'll be coordinated because of these gap junctions. Second method, contact-dependent signaling. So as this suggests, in order for this signaling to happen, two cells need to contact each other. They need to come in contact together. So what does this mean about the ligand? So far we've been talking about ligands that get secreted, released by one cell. So here's the receptor on the target cell, the cell that's going to respond. Here's the cell that's communicating. But if this is contact-dependent, what does that mean about this ligand? Well, it means the ligand has to be membrane-bound as well. So the ligand actually isn't released, it's a membrane protein, and so in order for this ligand to activate this receptor, the two cells have to come in contact. Right? So there's several examples. Contact inhibition for cell growth. So when a cell is crowded, it should not divide anymore. If it divides despite being crowded, that's part of cancer. Right? And so most cells, when they get crowded and they start to really contact each other, they're going to basically signal to each other, do not grow. Stop growing. That's contact dependent. Also, the immune system, we talked about this a little bit earlier, but T cell activation has to have contact dependent signaling with your cell. So this is just immunology 101. You don't have to know this, it's a little bit extra, but we will learn later on that DNA codes for proteins, but DNA gets transcribed into mRNA, mRNA gets made into protein. But most of these things are still inside the cell. So if we have a T cell coming around the outside and it's trying to figure out if this cell is infected or not, there's no way of it knowing because the T cell is outside the membrane. These things are inside the membrane. So what the cell does is it chops up a very small percentage of all the proteins it makes. It will chop up into these small peptides, and it will present them at a cell surface on a receptor called MHC. Right, so it's basically giving a small smattering of all the proteins it's making. Then the T cell comes along and it can bind, this is contact-dependent signaling, it binds to this MHC receptor along with that antigen. The antigen is this example of this small protein that's being presented at the cell surface. Now let's think about if your cell is infected with coronavirus, for example. Well, viruses hijack your cell. They use your cell to make some of its own DNA and make its own viral proteins. So if your cell is infected, it'll be making some viral proteins along with the normal proteins. And if that's the case, hopefully some of that viral proteins randomly get selected and loaded at the cell surface. This T cell now will recognize that as foreign. That is not something your body should be making. It's not part of your genome. It's a foreign genome. It's the virus genome. That's a viral protein. Now this T cell will recognize that this must be infected and will cause that cell to die. But this is contact-dependent signaling because the ligand that's causing signaling is bound to the T cell, and the receptor on both cells, both the ligand and receptor are membrane bound. That causes contact-dependent signaling. The two cells have to bind together to cause signaling. Then we have paracrine signaling. All right, so paracrine signaling is back to the ligand being secreted. All right, so this protein is made in one cell, it's secreted, it's released, and then adjacent cells will have receptors for it. And this is secreted through the secretory pathway that we talked about in one of the earlier chapters, chapter 4. And so that protein is made in the ER, it has signal sequences, it gets put in vesicles, and then those vesicles cause the ligand to be released outside of the cell, and then as it's released, it'll bind to adjacent cells' receptors, causing signaling. Signaling cell, responding cells. Okay. And so there's lots and lots of examples of this. An example is neurons at the synapse. At the synapse, one neuron will release neurotransmitters, and the next neuron binds those neurotransmitters, and that causes a signal to go from one cell to the next. But there's lots and lots of examples of this. In fact, most signaling is either paracrine or autocrine. And there's only a very subtle difference between paracrine and autocrine. So this is your signaling cell, and this ligand protein is being secreted, and it will bind and activate all cells that have the receptor. What do you think the difference between autocrine signaling and paracrine signaling is? So here's our paracrine signaling. Here's our autocrine signaling. Pay attention to the receptors. What cells have the receptors? What might be the difference? Well, the only difference is that in paracrine signaling, the cell that's releasing the signal does not respond to it because it doesn't have the receptors. In order to respond, you have to have the receptor for the ligand. Right? On the other hand, in autocrine signaling, the adjacent cells will respond because they have the receptor, but the cell that released the signal also has the receptors, so it will respond to its own signal as well. Right? So autocrine signaling, the cell that released the signal also responds in addition to the cells around it. All right, so there we go. Paracrine signaling only affects the cells around it. Autocrine signaling affects the cells around it and itself. And then finally we have endocrine signaling, and this is hormones traveling through the bloodstream. Right? And so this is back full circle to our adrenaline. Adrenaline travels through the bloodstream in order to affect all those different types. And different cells will have the receptor. Your hands have the receptor, but that receptor causes sweaty palms. Your eyes have the receptor, but that receptor causes dilation. Your liver has receptors. That receptor causes release of glucose. So these cells might have different receptors, all that respond to the same ligand. But not all your body, not every cell in your body responds to adrenaline. That would be really weird. Let's say adrenaline caused all your cells to react. Well, then your toenails will start growing if you have an adrenaline spike, right? Your hair will start growing. Everything that your body does will happen due to adrenaline, but that doesn't occur. Why don't all the cells react to endocrine signal? Well, it's because not all the cells have the same receptor. The adrenaline is in the bloodstream, and pretty much bloodstream means it has access to it throughout your body, but not all parts of your body will have the receptor. No receptor, no response. Why are there different thresholds to that same adrenaline? Well, again, full circle. Different thresholds because some of those receptors have higher affinity. Higher affinity means they have a lower threshold. Lower affinity receptors have a higher threshold. Also, some cells might have more receptors. More receptors, lower threshold. So the difference, a cell has to have the receptor to signal, And depending on the affinity and the number of receptors will determine how high or low that threshold is. All right, so what I want you to do now is basically kind of summarize what we've just talked about. So let's say that dry mouth and sweaty palms occurs with the least amount of adrenaline. Why? So see if you can write down in your own words why not all cells in your body respond to adrenaline and why the dry mouth and the sweaty palms is a response to relatively low amounts of adrenaline, whereas things like blood vessel constriction, heart rate, is higher levels of adrenaline. All right. Hopefully you were able to do that. This is kind of just a practice for exams, for example, because when we ask you things on exam, you have to be able to explain it to us and show us you know it. Well, here's the answer. Cellular response, it depends on the receptor. Does the cell have a receptor for the ligand? If it does not have the receptor, it cannot respond. Then what type of receptor does it have? Right? So depending on the number of receptors and the affinities, you're going to get different thresholds. Right? So the response is dependent on the receptor. You can have a ligand, but if there's no receptor, there's nothing to respond to that ligand. Right? And this is one of the major reasons adrenaline has different effects on different parts of the body. So, all right, the 3 things you need to take away from this asynchronous video. 1, you need to know the 3 major parts of cell signaling. We're going to get into those detailed examples of those later on. But the 3 parts, ligand binding to receptor and receptor has a conformation change. Signaling cascade, all right, signal transduction, and then the cellular response. All right, so that's one of them, those 3 steps. A second one is the idea of threshold. And we now know that more receptors, lower threshold. Higher affinity, lower threshold. But you should understand what threshold means and kind of how that relates to everything with the example of the adrenaline that we used. And then finally, you need to know the 5 major methods of signaling that we went through. All right, so we have intercellular signaling with the gap junctions. We have direct contact signaling, or contact-dependent signaling as it's often called. We have autocrine signaling, paracrine signaling, and then we have endocrine signaling with the hormones. All right, come to us and to the review session with questions, and we'll see you on Wednesday.